Matters much so. So the first speaker is Matt Gerhart, and he's going to be talking to us about conservation. He currently serves as the conservation director for the Northern Virginia Conservation Trust. He's been in that role since 2019. He protects the region's diverse natural, agricultural, scenic, and historical landscapes. Thanks. You very much.
Yeah.
He has over 20 years of experience in land conservation policy and practice. He's previously led those efforts in the San Francisco Bay Area Conservancy Program and the California State Coastal Conservancy. So West Coast.
I had a long haul out there. Yep.
And he holds a master's degree in geography from the University of California at Berkeley and a bachelor's from Yale University. And thank you for being here, sir.
Thank you. Thanks, everyone.
Get started. Can I just say a couple words?
Please, yes.
With sort of history on the land conservation front, and I apologize, I forgot to print this. But DWSP membership has traditionally placed a very high value on forest cover and riparian buffers that protect water quality. And so over the past several years, DWSP has, and even before that, DWSP has occasionally met with different land conservation groups, including Matt's group, the Northern Virginia Conservation Trust, to explore areas where there might be common ground between DWSP and also apply your drinking water and source water protection expertise. And so just to find intersections essentially. And so the idea of a mapping tool emerged, and we, DWSP which is not what Matt's going to talk about, but we did over the last couple of years develop our own land prioritization tool. And the goal, one of the goals was to help local and state government agencies and also conservation groups have a map layer that we feel as a DWSP would be helpful in terms of to protect drinking water quality. And so a subset of DWSP utilities, I believe 6, co-funded that effort. ICPRB was the contractor, and then we had a matching support from the U.S. Endowment for Forestry and Communities. And so that project was completed in around 2020, 2021, and has since been rolled out. The meeting that we're having now here at this facility, we're in a very urbanized and highly used Occoquan subwatershed. So this area serves as the primary water supply for Fairfax Water's Griffith Water Treatment Plant. And so Matt is going to talk about some work that they've done in conjunction with Fairfax Water to prioritize land here and to help protect the Occoquan Reservoir. So thank you for letting me interject.
Thank you. Yeah, and it was great to participate a bit in that effort and see some of the alignments between the way we approach things and the way the water agencies are thinking about it now. I'm going to try to describe as quickly as I can a sort of broad trajectory, and hopefully that spurs some thoughts or conversation. Again, I'll just go a little bit about us as an organization in case you're not familiar with Northern Virginia Conservation Trust or land trusts in general. Talk for a second about conservation easements, which are our primary tool for doing protection in the area. Touch on the Occoquan pilot that we worked with Fairfax Water on, and then our most recent effort has been to expand that to our service area, sort of a region-wide analysis for our use, and then also in working with partners where it makes sense, we can share what we've been finding. A little bit about the Northern Virginia Conservation Trust. Happy to have Mike Nardililli, one of our former directors here. We are going to be, I think, 30 years old as of next year as a regional land trust. We started out in Fairfax County as a pretty micro scale land trust, but eventually have grown to cover the region, which is, for us, it's a 2.1 million acre service area and 3 million people, and we have 9 staff. So we're kind of a small but mighty effort, which is not uncommon for land trusts. There's some very large national land conservation groups. Many land trusts have a volunteer kitchen table board, and everything in between. We're sort of a medium-sized organization. That's our service area. Shows sort of, I'd call it Northern Virginia plus. It's not quite just Northern Virginia anymore, but most of our effort has been in sort of the Fairfax-Loudoun corridor down towards here. We have formal partnerships with a number of the counties, including Fairfax, Arlington, and Alexandria. And most recently, as of last year, Stafford County has added sort of a working relationship with us with some dedicated staff for Stafford. We protected about 8,500 acres of land. It's about 160 sites. And the majority of that is through what we call conservation easements. Again, I'll do this super quick, but some people have never heard of this tool. We do work to purchase and then transfer land, or either own and manage it ourselves or give it to public entities. So traditional public land management is definitely part of the picture. The majority of what we do, though, is work with private landowners. The conservation easement is a permanent tool that essentially restricts certain uses on the land as agreed to between the landowner and the land trust, the holder, and that can be a government entity as well. And it has some key features in that it's a private landowner mechanism that is voluntary by the landowner. There's no regulatory component. It's permanent. These are in perpetuity restrictions that run with the land. And in Virginia, we have some very generous tax credits that help facilitate that work. So it doesn't take a lot of public dollars to lay out to get it done. That said, we have a very active land trust community. It's called Virginia's United Land Trust. You can look them up. Maybe 60-ish groups in the state. There's a lot of variability in how work has gone on in both Virginia as a whole, and specifically, if you I didn't zoom into our region, but you can see how much more there is in sort of our up watershed, Fauquier, Loudoun zone than a lot of where we have focused. That variability has a lot to do with the urbanization of our region and honestly just the engagement of communities with this kind of work. So what you see in these easements are these types of restrictions, and it's good to think about that for, I think, this venue in terms of where can a land use restriction on a private property relate to water quality. Mainly for us, it's in riparian buffers, protections on wetlands, and those kinds of things. We will, where say agricultural uses are involved, we will work with best management practices and things like that, so we can do some form of BMP implementation. But typically that's a little more on the voluntary side. Folks will work with NRCS to implement BMPs. So there's not a lot of, there's sort of an intersection, but it's not a complete water quality tool, I would say. One thing to think about. This is what we're trying to protect. And it's a huge, wide range of things. It's the simplest way to put it. We call them the conservation values that we're protecting. And part of the effort that what I want to convey is that part of our challenge is to be as focused as we can when we have so many different things that are out there that seem worth protecting. You'll see again the intersection here. So a little thematic version of what this is like for us. So I've always felt like we do have a lot of land trusts. We do have a target. We do have a bullseye. It just happens to be a really large bullseye. It's a little too large, right? And there may be an analogy to your work here too. And so what we've been trying to do through these processes is try to come up with the sort of areas of these different values, the tangible things we want to protect, and create a tighter circle there where we really are focusing in on multiple benefits at once for multiple purposes. And the rest of this, I'll just dive as quickly as I can into the actual data analysis, which I'm happy to share with folks or discuss. Just some reminders, as we launched into this, it's about a 5-year effort since our pilot. You get out what you put in terms of data quality, in terms of the structure of the data, and a lot of the work had to go in up front to look at how could we get at the diversity of landscapes in our region. It's such a diverse region. I have 1/10 of an acre protected in Arlington, and I have 450 acres protected out here. And that scale is really hard to work with in a landscape setting. So we started off in around 2019-20, with generous funding from Fairfax Water to look at the Occoquan as one of our critical water source areas and an area that we know there's a lot of ongoing development going on. We've in fact, since this study, several of the places we prioritized they're gone. They are subdivisions. It's sad to say. We worked with the Chesapeake Conservancy and some other folks to assemble layers, and I'll show you sort of for our region-wide analysis what that looks like. Data layer cake sort of approach to what's out there, and we took our criteria most generously because we want to focus on what do as land conservation folks want to protect, but we kind of steered it towards water quality wherever we could. So we sort of tweaked the analysis towards water quality, came up with some areas that we felt like were really kind of core areas, but not trying to capture everything, trying to create connectivity and really let the data speak to us a bit about places we already worked. So that was our initial effort. It came to a head maybe in 2021, and we're pretty actively looking at opportunities throughout these blobs. I'll just call them blobs. Fancy word. We then worked with other funding, actually from the Land Trust Alliance and some climate funding we secured, to say, how can we do this region wide? And it's really hard. And I really, I laud the effort you all have done, because watching, I think we were doing the same thing at sort of the same time. And it does kind of make you pull your hair out. I'm not meaning to read this or have you digest it necessarily, but what we did is we took the same sort of approach with a regional scoring, but then our folks who helped us in this case is a team called Biohabitats out of Baltimore. We did some segmentation of the analysis based on urban zones and socially vulnerable zones. And in those areas, we kind of reprioritized the data. It wasn't different data necessarily, but we re-looked at it based on heat island effects, parks, other sorts of socioeconomic factors. And what the point of that was, is we couldn't have our urban areas competing with rural unless we had some way to kind of let things do as well as they can for what they are, if that makes sense. Compare apples to apples. So the segmentation allowed that to happen. And then we've assembled that into the maps that I'll show in a sec. So just a real quick, we had a lot there, 24 layers. You'll see the watershed protections area is what we had pulled from the pilot, and really the emphasis on that. It's a separate set of data, which you can just look at that if you want to, or if we want to. But then we threw in what we could for everything else. And there's lots of data quality issues around doing that at this scale, but I think we got a pretty good outcome. And that's what we ended up with, is a very messy map, but an interesting map, because what it does is it takes the criteria, adds them up. This is a parcel level analysis that we now are going to work into a viewer. It can give you up to 1,000 in theory, but we have between 400 and 800 scores across parcels or in the region. And you do start to see areas pop out in both river corridors in particular, and in some of our urban areas, which is really what we were hoping to get at. And so when you actually dive in a little deeper, you get some of that diversity that exists in our sort of rapidly suburbanizing areas, where you have really nice forests still sitting there, and then there's 37 new homes and a bunch of, dirty water running in the stream. You have that on the landscape, and we're seeing it in the model. So we did a lot of validation to see is this really kind of gut check? Is it the right answer, more or less? We tried to update for recent development. We're always behind with these tools by a lot, unfortunately. So then what we're trying to do, and this is kind of getting into now, what we're trying to do is we pull the top sort of tiers of that analysis, and we started to talk with community members and our partners and said, what matters to us? What kinds of areas come out of this analysis as the things we want to not just say the data say are important, but like how can we generate support and energy around these areas? A concept of what it is. So obviously that's not easy when you see that kind of spaghetti. And we looked at other examples. These are our two regional commissions plans from 2009 for Northern Virginia, and GWRC's analysis, which we helped participate in a while back. You can see though a very different approach, right? Really generic kind of big blobs, or hyper detailed GIS, and practically everything is something. So which is the strategic part of your plan? Is one of the questions I ask everybody. How strategic is your plan actually? And we wanted to get somewhere in between these two kind of zones, where you just kind of have big swaths and you don't even know what you're talking about, versus everything counts. So this is just a little bit of where we are with this. We've kind of narrowed it down to different types of areas, and around 28 region-wide, which is still too many for us to focus on. But we are able to now look at them and kind of characterize them in different ways, and really talk about what it is that matters in those areas. So this little diagram is sort of depicting only that we have sort of areas that are geographic, and Pope's Head Creek, which is right up there, is a large intact corridor. It needs to be protected. But the river corridors themselves end up being the network, especially for this part of the region. When you're out farther, it's the ag land corridors, and they have a different character. So what we can start to do is characterize that quality and ideally start building campaigns around different protection efforts. So that's kind of where we are. And we are working on some strategic planning. We are looking to engage with partners in the region. So if you ever have feedback and you want to get into this data with us, let me know. And happy to answer any questions. Based on who it is? Well, we did on the basis of private versus public. So though all that screens out is what's considered protected, and that's actually not even a layer that exists that I can just hand you. There's actually lots of inconsistencies in people's protected layers, but in general, public land was taken off the table. There are some layers that do what's called gap analysis and sort of discount. I mean, Quantico is shown as protected in these layers. Is Quantico protected? You know, it sort of depends on who you talk to. So that's mainly our focus has been with private landowners who are farmers, ranchers, and actually residential owners. I mean, we work quite a bit with just people who have a house and 25 acres, you know, and that's really, there's still a lot to be gained by protecting that land. Yeah.
Really fascinating. I haven't been very aware of all this work you guys have been doing. Is this available on your website, or some of these products available on your website, or where would we find them?
Not quite, and it's a similar thing to, I think, your own analysis, in that we want the word out that we're doing this and we're doing it in a strategic way. But what land groups don't typically want to do is put out the maps that put, you know, bullseyes on people's properties, because then people, I'll tell you what you do when you have a web tool. You go to your house. It's the very first thing you check, and honestly, you look at it and you say, "Oh, it's totally junk. Like, it didn't say I'm important, you know, or it did." Or, so the honest truth is, we're happy to share kind of through a tacit partnerships, but we're not going to have a sort of blanket public view of this. It's really an internal plan for.
I really wasn't asking that level of detail.
Oh, sure, no problem. Oh, sure. We don't have it yet, but actually, I'm happy to produce something like that. Yeah, in fact, this is about the closest we've gotten to a summary.
I know the field.
Yeah. But yeah, definitely happy to talk that. And actually, an area that I think might be interesting is to compare. I know our analyses don't actually cover the same geography quite, and you focus a little further upstream, but to just see what pops out in each one independently, I think would be a really interesting exercise, especially for Loudoun, you know, and particularly, and parts of Fairfax. I think we could learn something there. All right, thank you.
Thank you. Next up, we have Dr. Jones, who's going to talk about toxic cyanobacteria, benthic harmful algal blooms, and the Shenandoah River. So Dr. Jones is founder and director of the Potomac Environmental Research and Education Center. Thank you. And as you know, it's located on the title of the Algal River here in Woodbridge. PERIC is home to 8 faculty researchers from George Mason University, with the College of Science, and 3 from GMU's College of Engineering and Computing. He's been a researcher since 1980, founded the long-term ecological study of Gunston Cove, which has been conducted annually since 1984. You've mentored over 20 PhD students, 40 MS thesis students, and has been awarded the Jack Wood Award for the Town Gown Relations and the Earl C. Williams Presidential Medal for Excellence in Social Impact at GMU. Thank you very much, sir.
Okay. They wanted a bio, so you produce the best you can. So pleasure to be here, and a super pleasure to have you guys here. We really value all the partners that come and participate here. Sometimes we're relevant and we come down and talk. Sometimes we're not, but that's okay. We love to host you guys, and beautiful view of the tidal Occoquan there. So, no, it's a little further downstream than most of your activities, but we're all connected, right? So, at any rate, yeah, as was stated, I'm my historically, and there's a lot of history there, right? My emphasis has been in the tidal Potomac, but I have done some work in the upper watershed. But recently, in the last year, we have become involved in the Shenandoah, which is a really another place that's a site for sore ops, right? It's a great place to work. And so I'm excited. I'm also excited that we're opening a new front on our research, which is toxic HAB blooms. And in particular, in the Potomac, upper Potomac watershed. So we know there's toxic blooms in estuaries and marine systems, dinoflagellates that have been studied for some time, but more recently, we're recognizing that stream systems, particularly shallow stream systems, can have major problems. So with that preamble, let me talk about a study that we are the analytical lab for. So just want to make that clear that we're not doing the field sampling for, but we're of course important as the analytical lab. So it's helping to build our capacity here at PERC. And so without further ado, and you guys are many of you know this quite well, but the Shenandoah River, of course, is the major tributary of the Potomac River. And it's composed of, what's the pointer here? Yeah, okay. It starts at Harper's Ferry, and it's one river here, and then it splits at Front Royal and becomes two major separate watersheds, the North Fork and the South Fork. And the South Fork runs all the way up here into, they both start more or less in the slopes of the, I guess I call it North Mountain, that runs between Virginia and West Virginia. But the majority of the drainage is actually in the valley. Okay. So if you were to say, I mean, I find it interesting, it's kind of digression, but I find it interesting that probably the South Fork should just be called the Shenandoah River, right? Then up here, maybe split it out. But yeah, that's the tradition. It must be fairly similar in discharge to get that. The North Fork is a little shorter, but both of them essentially drain similar area. So the land use, which I'm sure most of you are aware, is predominantly forest, actually, if you look at the whole watershed as a whole. But there's a sizable amount of agricultural land use, all the yellow, and there's also some orange in here, which is kind of hard to see, but like right in here is mostly orange. So it's a combination of pasture lands and cultivated crops, and that's what generally adjoins the river, except maybe for a very narrow strip of forest buffer. So arguably that would have a large effect on water quality and on what goes on in the river. There are some small cities in the watersheds also shown. Okay, so there's two basic industries in this watershed. One is recreation. Of course, we have here kayakers. If you drive out there, there's all sorts of outfitters for kayaking, for canoeing, for fishing. Very active outdoor recreation area, especially in the summer. And one of the reasons is that the bedrock is fairly close to the surface here, so you get a very wide and fairly shallow water body. And that actually is, in many ways, very good for these activities. The other major land use, of course, is farming. And this is actually a painting that I own. It's kind of hard to see here, but I think it really captures the, there's no animals here, so we're not looking at cows, but you know they're there somewhere, and a little bit of cropping and so forth. And here's your stream draining that landscape. And very bucolic, but on the other hand, you see the close interaction between farming activities and streams. Not to draw a direct correlation between agriculture and toxic algal blooms, but just to say that they're related in some way. Okay, so filamentous algal blooms have plagued the Shenandoah River for at least a decade. I'm not able to go back much further and see stories in the newspaper, but at least a decade. These growths have rendered recreational activities more difficult and certainly less appealing. So here's, this was from Potomac Waterkeeper Network, but you could see this does not look like a fun place to kayak, right? And fishing would be difficult, right? You throw your line in and you immediately have to reel in all the algae. So these were not desirable, but people were just kind of dealing with them. You know, the outfitters would try to go to places that weren't clogged up, and kayakers and canoeists would deal with it because they weren't toxic. Okay, but that all changed in 2021 when there were significant growths of benthic cyanobacteria, which commonly known as blue-green algae. And, you know, the reason why they're lumped in with algae is because in terms of their basic ecology and their occurrence, they co-occur with eukaryotic algae. Okay, so they tend to get studied more with algae than with bacteria, which they're more closely related in a molecular way, let's say. So anyway, that's a digression, but not surprisingly, if you have benthic green algae, you're probably going to have some benthic cyanobacteria. In this case, the benthic cyanobacteria kind of took over the system, and they were found in the North Fork. Remember, that's the somewhat smaller one, which is really the West Fork in some ways, west of the South Fork. And then some, the health department got into, I think BDH got involved and did some toxin measurements, and they actually found measurable levels of toxins in the North Fork. And they issued a health advisory cautioning the public to avoid water contact and within the parts of the North Fork, and they eventually extended it 53 miles, which effectively excluded the North Fork from recreation. That's almost the whole part of the North Fork that would normally be used for recreation. There were also water takes involved. Then there was a subsequent smaller bloom in 2022, which looked like it was going to be worse maybe, but then we had a major flow event that kind of flushed it out. But during this process, the political leaders in Shenandoah Valley said we're under pressure from the public, who were actually some of the outfitters and so forth, to do something about this, not to mention the people that wanted to recreate there. So the General Assembly, Virginia, passed legislation to fund studies of the blooms in the Shenandoah River and also in Lake Anna, which had been subject to many algal blooms as well, cyanobacteria blooms. So once the legislature appropriated the money, they turned it over to DEQ, Virginia DEQ, who then contracted with USGS and ICPRB to do the studies. Neither one of those entities had a lab. that could, that was interested or could deal with this, the volume of samples and the type of samples. Originally, they looked to VCU, but that didn't work out. So fortunately, from my point of view, they found us and we found them. And so we've got a contract going. And we've been given samples this year, and the study will continue through actually June 30th of 2025, but the last samples will be collected at the end of 2024. So we got kind of a partial year of samples this year due to the late start, delay that's often happens with contracting and so forth. So this, unfortunately, we can't really block that light up. So that side of the projection seems to be difficult. But this is the North Fork. So this map is turned so that the North Fork is now totally a West Fork, right? So north, actually over here somewhere. But anyway, that's the North Fork. Here's the South Fork. This is the whole section basically that was closed on the North Fork. So we have multiple sampling sites there. There's 4 stations a little farther up on the South Fork. The South Fork gets too deep down in the lower range. So this is more analogous section of the South Fork. And partially that was for kind of have a reference area, but we're finding the same HABs now in the South Fork. So it's good that we're measuring there. So I'll talk a little bit in a minute about the way we're sampling the mats, but also USGS is collecting water column samples basically above and below each of these groups of mat bottom sampling areas. And those are existing USGS sites. And the idea there is to look at what may be coming down in the water after and during these blooms. So is some of this material not just staying on the bottom, but actually being transported downstream, which could have some implications on downstream water intakes. Okay, so just to give you a quick idea, and this, we took our sample design from a California protocol, which fortunately one of our team members was involved in before she left California and moved here this year. So in this case, we're doing quantitative sampling of algal mats. So at each site, we have multiple transects, and at each transect, we either collect from one shore, random point from one shallow area on the right shore, then go to the left shore on the next transect, then go to the middle on the next transect. We do that 3 times. So we end up with this total of 9 subsamples. They all get combined together because the analysis of this stuff is, time consuming, and also it's very spotty. So it's better just go ahead and say we're going to take 9 samples and it's going to be an average of the habitat rather than trying to do them separately. So this is our composite sample, which basically just goes into a large container, mixed thoroughly. And what's happening now in California, they do this segmentation in the field, but we found that the field crews are very stressed just to get the sampling done. They're tired, and it's just the kind of thing that you don't really want to, make them do in the field. So these samples are coming back in the jugs to us, and then by the end of the day, then the next morning we come in and we do the segmentation into diatoms, what they call soft-bodied algae, which is everything but diatoms. Chlorophyll, we do ash-free mass, but also not shown here, we do toxin analysis, and we're doing molecular analysis too. Okay, let me just quickly, we do some qualitative samples as well. So if they see mats that are not sampled by this statistical process, they will grab those too. And we will do qualitative analysis, meaning looking to see what species are there. If there's blue-greens there, we may do toxin analysis on those. Well, and then as I mentioned, the water column samples USGS collects. And this is detail on the measurements that we make. Can't see this hardly at all, but this is a picture of a mat sort of from 3 or 4 feet away. And they're more distinguishable in that they're usually darker. And you can't see the filaments as well. They're finer in general filaments. So it just looks like a dark globs on the sediment surface. And this is what some of the representative taxa look like. This is this is a filament. Think of the individual cells like nickels stacked on top of each other. So once you get a stack of nickels, you get a filament. But they're mostly filamentous like this, filamentous colonies. And then there's several taxa, there's Lingia, Microcoleus, Anabina, and Oscillatoria are some of the taxa. So we're doing the taxonomic identification and enumerating all the filaments, including the green algal filaments. We're doing pigments, chlorophyll A, so that gives us a measure of algal biomass within these samples. So chlorophyll A is found in all algae, but phycocyanin is another pigment. It's only found in cyanobacteria. So this gets a relative amount of those two. Ash-free dry weight is just you take a sample, you dry it out, you weigh it, then you combust it, and you subtract dry weight from the ash weight. You get ash-free dry weight, or I would call it organic weight. Okay. So that gives you an idea of the total mass there. But if there's things, it's gonna include like decaying leaves and stuff like that. So it's a cruder measure of mass, but you use it to standardize a lot of things. Then 2 ways of measuring toxins. We're doing ELISA assays, which is an immunological assay that targets individual toxins, but it doesn't give you a lot of information about their quantities. And then we're following up on those positive results with LCMS assays, which allows you to quantify the toxin levels. And also, I got a separate grant, which happened to coincide with this, and we're using that from GMU. We're using that to do molecular analyses. So this is my team. Very proud of them all, including myself. And so we have chemists, and we have microbiologists. Dr. Christova is our mycologist expert, and the one from California. And I sort of try to keep the whole business moving. Many of you know ICPRB know Mike Seligman. He's our field, he heads up the field crew. So, and then Doug Chambers heads up the field crew for USGS. I think that's all I have. Yeah. Thank you.
Any questions for Dr. Jones?
Yes, sir.
We're increasingly getting concerned about antitoxin. A few reasons. That's the one that we don't have EPA health advisory level 4. So we are looking into different states in Hawaii, EPA, WHO, but we're basically looking for reliable health reference level. It presents unique treatment challenges. Earlier this year, there was a bloom upstream, not upstream, but just upstream of our intake, where they found super high level of antitoxin from the cell, but not in the water column. So we're trying to better understand how it behaves and the risk factors and how we should, respond to the events to mitigate the risks. So you earlier mentioned that you issued the health advisory. And I was wondering what were the criteria for those advisories and what were the levels, the trigger levels? And
well, that was Virginia Department of Health. So That was even before I got involved in this, but my feeling is that if they could detect toxin, they were going to issue a health advisory. And detect toxin and see the types of organisms that they, in significant amounts that they expected to produce toxin. What I was reviewing all this last night, trying to know the most definitive statement I could about that. And so I don't know if they were looking at toxin in the water or toxin in the cells. And the limited data that we have back now says that most of the toxin is in the cells. It hasn't been diffused or been excreted into the water. However, you can imagine if the bloom dies, everything that's in the cell is going to become liable for release. So it seems like if it's in the cells, you got to worry about it too. Yes.
So this year, flows in the Potomac have been low. And if we look at the drought maps available, it's clear that the Shenandoah River is probably the predominant reason. You know, we can debate whether it's climate change or not climate change that's causing this. But I guess, you know, even beyond just your question that you posed about could the mats, you know, transport downstream, I'm also curious, you know, can we see the same type of, you know, progression of algal activity in the Potomac?
Yeah, absolutely.
Because the other thing you talked about was, well, first you saw green algae and then we started seeing the cyanobacteria events, right? So I think there's a whole area potentially of exploration in the connectivity between all of this, right? The weather, no rain, there's calm, still environment, shallow river, right?
Yeah.
All of that kind of practical stuff.
Yeah.
That, I mean, we could be seeing the same thing.
Yeah.
In our stretch of Potomac.
Yeah, and I certainly the Potomac at Harper's Ferry looks exactly like the Shenandoah River, right? It's very wide, very shallow. I don't know how far down that type of topography goes, but that whole stretch seems to me like it's very vulnerable. And we would like to extend our studies into that area. We'd probably do a little bit of exploratory sampling. I don't have money for it, but we can, you know, do a little bit. Students can get involved. So I think it's a really good question and really good chance that these will extend into that part of the Potomac anyway. Because what happens is you get some of these things like to grow in the fast flowing, but a lot of them like to grow in the little pool areas. And the pool areas, like when you get down toward, I'm just going to say River Bend, you know, you get down toward the more closer to the falls, the pool areas are deep, but up there, the pool areas are shallow. So there's plenty of light hitting the bottom of the pool area, which also has some fine sediment, which has more nutrients in it. And so it's, that's where we find a lot of them. But some of them do actually need organic matter to grow.
Yeah, and I think Frederick County and Maryland DNR Fisheries and Kevin Seltner have done some work in that area of the Potomac, but I think it's been much more ad hoc than what you're describing, was much more systematic.
Yeah.
It would be, I think, really worthwhile to think about expanding the geographic scope of the sampling to see what's going on.
Yeah.
It's been, like you said, a problem for a few years. It's probably not going away.
Yeah.
I think there could be some benefit to that. I know Maryland DNR, their fisheries folks are out quite a bit. I think are able to like do sampling if they knew what to do.
Yeah.
So.
Yeah, we can, you know, this project's really enabling us to get up to this capacity on analytical work. I mean, we have the basic faculty, we have the basic, but, you know, we didn't have all the methods down, all this, we're learning a lot from other states. So we'll have all that together, and it'd be great to look at other places as well that are similar. Yeah.
Sounds like a good work group.
Thanks a lot.
Thank you.
Your first is up. She's an assistant professor of environmental engineering in the College of Engineering and Computing at George Mason University. She's an affiliate faculty in the Department of Chemistry and Biochemistry. Her research spans water and wastewater, and I believe she's going to be talking to us about some of that today. So thank you.
Thank you. I'm really glad to be here. Thank you, Christina, for the invitation. I think you all have the opportunity, I believe, to tour my lab upstairs after lunch. So just briefly, my research, as mentioned, has in the past focused on water reuse in particular, and looking at disinfection byproducts and other organic contaminants in the water reuse cycle. Really focused more on inside the fence, so to speak, on the treatment side and distribution side. But since joining Mason just in 2020, just a few years ago, I've started shifting some of my focus to outside of the fence, looking more specifically at source water. And that's partly driven by the interests of my colleagues like Chris here at the Potomac Science Center, as well as interests of my students, who you'll hopefully get to meet when you tour my lab. So yeah, I'll be talking today about trying to kind of broadly think about the potential impacts of a changing climate on the Potomac River Basin in terms of the disinfection byproduct precursors. And I'm coming at this with that lens of thinking about water reuse. And that includes intentional reuse as well as de facto reuse, which I'll define in a moment. But first, I'll start with disinfection byproducts for those who are uninitiated. It's a little bit more complex than your typical contaminant. And then I have more slides than time, so I'll gloss over some of this, but I believe Christina's sharing our slides after the talk, so if you're interested, you can take a look at that. And I'll talk about some of my previous work and some projects that we have starting that are related to this. So first, disinfection byproducts. We can't talk about them without talking about how important disinfection is. So the goal of drinking water disinfection is to protect against waterborne pathogens. And of the common disinfectants, chloramine is the one that's primarily used in our area. Other drinking water utilities in the basin surely use chlorine, which remains the most common, most popular disinfectant. And this was really a significant public health innovation, 130 years ago or so, and it has saved countless lives. But unfortunately, like so many good things, it has unintended consequences. And the big one for disinfectants are that they don't just react with pathogens, they're nonspecific. They react with basically anything that can be oxidized. So that includes organic matter, natural as well as anthropogenic. And salts like bromide and iodide also get involved in these reactions and change the speciation of disinfection byproducts. And inorganic nitrogen and some other constituents can also get involved. So the source water quality, as well as what's removed in the treatment train, will determine what disinfection byproducts are formed. But basically, in every single water, there will be some mixture of disinfection byproducts, some of which are known, some of which are potentially still unknown to us. And so there are over 700 species that have been identified. Most of these are not measured in any given study. Typically the regulated species are used as indicators. And we actually don't know which cause disease. We just know that disinfection byproduct exposure is strongly associated with particularly bladder cancer and a number of reproductive health effects. So this is, important to balance the risk of pathogen exposure with DBPs, with the balance always tilted towards preventing pathogen exposure. So some regulated DBPs here, quick introduction. We have trihalomethanes, which are the most commonly measured around the world and regulated around the world. These are regulated in the United States specifically, by the way. So we have these 4 THMs and then 5 HAs and a couple of inorganic DBPs, which tend to be rather specific. And then there's all this vast universe of unregulated DBPs. The ones that I will focus on the most here are several classes that are of potential regulatory interest. Either the field has been pushing EPA to consider regulating them, or EPA is actively considering regulating them. So halocyanonitriles are really in the former category. We believe they're very toxic. That's what the toxicologists tell us. And they show up in basically every water that has even a little bit of organic nitrogen. So they're pretty common. And then there's nitrosamines and nitrosodimethylamine being the most common one and of concern for water reuse systems. And then the brominated haloacetic acids. So these 5 plus these 4 here together make HAA9. So 4 of the brominated haloacetic acids are not regulated. This, could be a concern for water supplies that have high bromide. If EPA, as they're considering, regulates HA9 instead of HA5, then this could mean that systems that have high brominated DBP formation, could be affected by those new regulations if it happens. Just quickly, the brominated DBPs are more toxic than the chlorinated DBPs. And the nitrogen-containing DBPs are typically more toxic than the carbonaceous DBPs. And we know this from a variety of toxicology evidence, but particularly this database at the University of Illinois, where they have done this assay with mammalian cells, these Chinese hamster ovary cells, to look at both broad screen toxicology, so chronic cytotoxicity, which covers a lot of different endpoints, as well as genotoxicity. And these trends are pretty consistent across that database, as well as the broader literature. So with that in mind, this is from a paper led by Rich Weissman, who's EPA scientist and until recently a PhD student at Mason, mostly before my time, and I showed up in time to collaborate on this work. So he looked at this UCMR4 database, which a lot of you are probably familiar with, the EPA Unregulated Contaminant Monitoring Rule 4, where they required that all large water systems across the country measure DBP precursor levels at their intakes, which they're already doing, at least for TOC, and report HA levels from their systems. So this is just very, very broad, only looking at surface water source intake levels of TOC, total organic carbon, and bromide by EPA region. So really blurring our eyes here, but you can see the Potomac River Basin, I believe, fits entirely within 6, 5, and 2. It's what it kind of looks like on the map for the most part. So we're enjoying low bromide and low TOC in this area, which is great. So the question that I've been thinking about is that going to change with the trends that we're seeing in relation to climate and extreme weather? So here are some of the factors related to climate and extreme weather that could affect the loading of DBP precursors in the basin. And I think the first one that is probably on a lot of our minds right now is drought. Lower water levels, less dilution of pollution, right? And then extreme precipitation is something I've been thinking about as well, and I'll kind of come around to that at the end. Algal blooms, we just heard quite a lot about, and I won't talk about that except just to say that they do tend to add a lot of nitrogen, organic nitrogen. So you tend to see a lot of halocyanonitriles downstream of algal blooms, which could be a regulatory concern and is definitely a concern for human health. And then heat can change the composition of organic matter. Probably a lot more. We could surely brainstorm a lot more. I like this diagram, which kind of outlines some of these impacts on a variety of contaminants. So it took me a bit I was looking at the ICPRB's drought monitoring page yesterday and looking at this graph where I was like, what am I looking at? Is this, I can't believe that was the flow yesterday. It's so low compared to historic levels. So, this really could have an impact on the concentration of a lot of things in the surface water, right? And particularly if we think about like municipal and industrial wastewater discharges, it means that they're getting less diluted. So, so that brings me to de facto reuse, which maybe you're all familiar with. This is the unintentional reuse of wastewater, basically an intake that just happens to be downstream from a wastewater discharge. Usually we think about full-scale wastewater treatment plants when we think about this and an actual outfall. I'm going to make an argument that there are other types of de facto reuse that we should probably be considering. And de facto reuse is very widespread. It's certainly happening throughout the Potomac River Basin. This is a kind of an overwhelming figure, but I like it because it just shows how widespread de facto reuse is throughout the U.S. And you can kind of see in the Potomac River Basin some of the potential hotspots, including right here in Northern Virginia, that big green circle is showing that on average back in 2015 and prior, the percentage of wastewater in the river at that point was 10 to 15%. And given the trends in the drought, we would expect that percentage is probably higher right now. And that's part of the reason, if you look at areas with low flow like Southern Texas, there's quite a lot of a high percentage of wastewater. And in the summer, in the dry season, I've heard it can be up past like 90% wastewater in rivers in Texas. So hopefully that's not our future. So just quickly, the percentage of de facto reuse, it affects DBP precursor levels. It contributes to TOC at the top. This is also work from Rich, and I guess I did not get the citation on here, so I'll follow up with that correction. Maybe it's on the next page. But this is the Shenandoah watershed. A lot of Rich's work focused on the Shenandoah watershed and de facto reuse and the impact on DBPs. And so the top graph is showing the TOC variability at a public water system intake in the Shenandoah watershed. And he found that was related to the percent of de facto reuse, which was basically estimated by just looking at the flow. And there is a relationship with THM formation and HA formation. These are the two regulated organic classes of DBPs. And the relationship is a little bit rough. There's so many factors that go into DBP formation, but it seems to be there across this data set. So something that is really well established with de facto reuse is, as I mentioned, or maybe I don't remember if I mentioned, maybe I didn't. So wastewater has a lot of organic nitrogen. You think of proteins, breakdown products of proteins and urea, et cetera. That's all organic nitrogen, and it is strongly associated with increasing levels of NDBPs, including those halocyanonitriles and NDMA. It also has more bromide than ambient surface water. So especially at usually in a healthy large flow river, you wouldn't necessarily see a noticeable increase in bromide, but at low flow you really might. So I'm going to kind of gloss over the next few slides. I wanted to show some actual DBP data from my research, which the field work that I've done is actually focused in South Asia, where de facto reuse is extremely prevalent. So just to quickly say that one of the ways that we can actually measure de facto reuse, or prove that it's happening in an area where we don't have good estimates for the wastewater volume that's being discharged, et cetera, is that we can measure tracers like sucralose, for example, which is fairly conservative in that it has a half-life of like 100 years. That's by design, right? That's why it's 0 calories, because we can't digest it. So it doesn't get digested in wastewater either, for the most part. And it's very water soluble, so it's a good indicator of wastewater. And we found that it was just ubiquitous across this northern state of Rajasthan in India and in the area of Dhaka and Bangladesh. So I'll leave that for your reading later. Basically the surface water that was impacted by wastewater looked really similar to some actual disinfected wastewater from California from another of my my papers. So and this is the cumulative calculated toxicity. So that was fairly alarming to us because these were actually samples of drinking water, of potable water in the distribution system in these cities in South Asia. So really quickly, I alluded to this, that I think there's other sources of wastewater that may particularly become important when we think about these climate extremes like drought and extreme precipitation. So I was pretty surprised to learn that there are 20,000 septic systems across Northern Virginia. And most of them are out in the western kind of more peri-urban rural areas, but there still are quite a lot sprinkled even throughout these fairly developed areas. So I think that's, surprising to me and a lot of other people. What's also surprising is that a lot of these are in our drinking watersheds. So this is the Occoquan Reservoir right here. We're like right here. And the dam is just a little bit upstream, and then this is the reservoir. And so a little under half of the septic systems in Fairfax County, not even counting what's in the other counties on the other side, are in the Occoquan watershed. And then Difficult Run has the majority of septic systems in the area, and then also Pond Branch and Nickel Run, so draining to the Potomac up there at the top. And it turns out that our soil is not great for septic systems. It's really poor in terms of organic carbon, which is one of the things that you need to encourage the microbes to actually eat the waste in septic fields, right? Or if it breaks through your leach field. And then we also get these extreme rain events that potentially just flush the entire landscape, it seems, right? And I know that in my yard, I get puddles everywhere when it rains, even kind of a moderate amount. And so good thing, I don't have a septic system, because I would definitely not trust that it was working well. And so we've done some studies. We did a limited range of preliminary data collection. Here's an example of one of the sites that we went to. This is on the Occoquan Reservoir. It's Ryan's Dam, an entirely septic sewer sheds and no sewers, just septic systems. And up here in this tiny little stream, we found a kind of surprising amount of pharmaceuticals. This is like a little snapshot of what we found, and the concentration of these pharmaceuticals was higher than in the reservoir in the main stem off the bank there. And we also found PFAS in that stream draining. Again, the only source that we're aware of is septic systems. So this was really surprising to us. And we're anticipating, this has not been publicly announced, so please don't repeat it, but I believe that we are getting a grant from USGS to look at this, to collaborate with the Virginia and West Virginia USGS Water Research Center, and really zero in on the impact of septic systems on drinking water quality on the Occoquan and the Potomac River side, and particularly looking at the effect of extreme precipitation and how much is that exacerbating contaminant breakthrough from all these septic systems. And my PhD student, Meghana, is leading that, and I think you'll meet her later if you visit our lab. So that's it. A lot of the work from earlier in the talk was actually from my graduate research at Stanford. So acknowledging my lab and the students at Stanford that helped me with that and our funding, and then the Basin Center for Resilient and Sustainable Communities, which funded our preliminary data collection out in the Fairfax County. So thank you for your attention, and I'm happy to take questions. I hope I didn't go over.
No, you did not. You had extra time. So.
Yeah.